Good morning, everyone. Today I will be discussing Ziehl, Neelsen staining, and cold Zn staining, focusing on their principles, procedures, and diagnostic applications. These techniques are used to identify acid-fast organisms, especially Mycobacterium species. Ziehl-Neelsen and cold variants are foundational acid-fast methods for detecting mycobacteria in routine microbiology workflows. Acid-fast principle. These methods exploit the unusual lipid barrier of acid-fast organisms, which retains dye despite harsh decolorization. Mycobacteria detection. They remain central for identifying mycobacteria in direct smears, especially when culture is slow or unavailable. Heat versus cold. Classical staining uses heat to drive dye uptake, whereas cold variants rely on chemical intensification without heating. Clinical relevance. In pulmonary and extrapulmonary specimens, these stains provide rapid, actionable information for infection control and triage. Principle of Ziehl-Neelsen staining. The Ziehl-Neelsen principle is built on lipid-rich cell walls, dye penetration, and selective retention after acid-alcohol treatment. Mycolic barrier. Mycobacteria possess waxy mycolic acids that create a hydrophobic envelope resistant to routine aqueous stains. Primary dye entry. Carbol-fuchsin, aided by phenolic formulation and heat, penetrates this barrier and binds deeply to the cell wall. Retention after decolorization. Acid alcohol removes stain from non-acid-fast cells, but not from organisms with strong lipid-rich envelopes. Contrast staining. A counterstain such as methylene blue or malachite green renders the background visible and improves recognition. Classical Zn stain workflow stepwise laboratory procedure. Classical staining follows a tightly ordered sequence, where each step prepares the smear for reliable acid-fast visualization. Prepare a thin, even smear and allow it to air dry completely before fixation. Heat step. Apply controlled heat during carbol-fuchsin staining to enhance dye penetration into acid-fast organisms. Decolorize. Use acid alcohol until excess primary stain is removed from non-resistant cells. Read slide. Examine under oil immersion for characteristic red bacilli against a blue or green background. Cold Zn stain. Procedure and rationale. Cold Ziehl-Neelsen methods remove the heating stage, trading thermal enhancement for safer and simpler bench execution. No heat. Eliminating flame or hot plates reduces procedural complexity and improves standardization at the bench. Chemical intensification. Alternative mordant systems support dye uptake and retention without relying on thermal assistance. Lower aerosol risk. Avoiding heating decreases splatter and aerosol generation, supporting safer manipulation of infectious material. Simpler handling. The workflow is easier to train, monitor, and reproduce in laboratories with limited infrastructure. Microscopic interpretation and reporting. Interpretation depends on morphology, color, distribution, and a consistent reporting scale that communicates organism burden clearly. Bacillary shape. Acid-fast bacilli appear as slender, slightly curved rods, often singly or in small clumps. Typical color. True positives usually stain bright red or fuchsia and stand out against the counterstained background. Result pattern. A positive smear shows discrete red bacilli. A negative smear lacks convincing acid-fast forms in examined fields. Grading scale. Semi-quantitative reporting translates observed bacillary density into standardized categories for clinical communication. Applications, limitations, and quality control. Quality and clinical utility depend on specimen context, reagent integrity, and disciplined internal controls throughout the process. Sample scope. Useful for pulmonary and extrapulmonary specimens, including sputum, tissue, aspirates, and body fluids. Error sources. Improper decolorization, debris, and weak bacillary loads can generate misleading positive or negative results. Reagent stability. Fresh, properly stored carbol-fuchsin and decolorizer are essential for reproducible staining performance. Quality control. Control slides and consistent technique help verify stain integrity and reveal drift in daily practice. To conclude, acid-fast staining, including both Ziehl, Neelsen, and cold Zn methods, remains a critical technique in microbiology. These methods allow rapid and reliable detection of acid-fast organisms, especially Mycobacterium tuberculosis, which is essential for early diagnosis and treatment. While the Ziehl Neelsen method uses heat to enhance dye penetration, the cold ZN method offers a safer and more convenient alternative without compromising diagnostic value. Ultimately, the accuracy of results depends not just on the method used, but on proper technique, timing, and careful interpretation. Thank you. If you found this video helpful, don't forget to subscribe to Meds.Concepts. Press the bell icon so you never miss an update. Share this video with your classmates and help them learn better. Let us know in the comments what topic you would like to see next. Keep learning, stay curious, and see you in the next video.